Hey everybody, how's it going? Today's video is going to cover protein import into the mammalian mitochondrial matrix. Now, again, as always, if you like the video, if you actually learned something from it, if it's useful to you, please go ahead and follow. It takes a while to make these videos, but I try to make them as useful as possible for you guys. Now, why do I say specifically to the mammalian mitochondrial matrix, right? Well, because we're interested in mammals here, right? Usually most textbooks and literature you read are going to be related to fungal or fungi or any other sort of bacteria, but the problem is that's not really useful to us, okay? So we want to learn about the mammals, all right? And I say specifically to the mitochondrial matrix because, there's multiple pathways that we can talk about, but this video is only going to be on the mitochondrial matrix, okay? So before I get into this, I have to give you guys an overview, okay? So here's the mitochondria, right? As you guys know, mitochondria, best known for its powerhouse abilities, it makes ATP, right? But it does so much more, and it requires many proteins, right? The electron transport chain, for example, that is studded along all of these little cristae, okay? We call it the folds in the mitochondria, cristae. The electron transport chain is made up of a whole bunch of proteins, and only some of those proteins are coded by the mitochondria itself. And if you are confused here, I'll tell you something right now. The mitochondria has DNA. Yes, it does. It has its own DNA. And it's the circular-looking DNA, okay? It's called mitochondrial DNA. It's circular, double-stranded DNA, okay? And this is actually a remainder from back in the day where the mitochondria was bacteria, specifically a purple sulfur bacteria. Purple sulfur bacteria, okay? And it evolved throughout that endosymbiotic theory, as you guys very well know. Now, this mitochondrial DNA, like I said, can only code for 13 proteins, which are required for respiration, right? The rest of the proteins, about 1,200 actually, or 99% of the proteins, come from the nuclear DNA. Okay, so they come from the nucleus of the cell. Now, if you guys remember basic biology, you're going to have a gene in the nucleus. That gene's going to get transcribed, okay? And then it'll give you an mRNA molecule, which will be translated in the cytosol. In this specific pathway, it'll be a free cytosolic ribosome. So a free ribosome, rather than the ribosomes that are attached to the endoplasmic reticulum. All right, it'll actually be a free cytosolic ribosome. And then that protein that we make from translation will get imported into the mitochondria. Notice how I have two channels here, okay? This blue one is going to be sitting on the inner membrane of the mitochondria, so it's going to be called TIM. TIM standing for the translocase. Okay, so the translocase of the inner mitochondrial membrane. Okay, translocase of the inner mitochondrial membrane. And this guy is on the outer membrane, so he's called TOM. Translocase of the outer mitochondrial membrane. Okay, that's great. Now, like I said, we're going to take this gene, okay, and we're going to grab some sort of enzyme. In this case, it's RNA polymerase. And it's going to transcribe this gene. When you see this symbol, it usually stands for transcription start site. It's going to transcribe this gene, make an mRNA. This mRNA will go through the nucleus, out into the cytosol. It will have gone through 5' capping and 3' adenylation to ensure mRNA stability. And then this mRNA will get translated into a protein. Now, since this protein is going to get imported into the mitochondria, we actually call this a precursor, okay? So a precursor protein. And on top of this protein, okay, on the N matrix, N-terminal side, sorry, it'll actually have what's known as a targeting signal. This is basically just a sequence of amino acids that tell this protein, hey, you're destined for the matrix of the mitochondria, okay? You're destined for the matrix of the mitochondria. Now, just a little, basic biology here. Here's a protein, okay? It's going to have a COH, so it's going to have a carboxyl end, and an amino end, okay? The amino end of the protein will be called an N-terminus, and this carboxy end will be called a C-terminus, okay? This terminology is going to be important in about a second, okay? So let's move on. If we were to actually, let me go back. If I were to take this picture and I were to zoom in on it, this is what I would see. So here's the cytosol. Here's the outer mitochondrial membrane. Here's the intermembrane space. Here's the inner mitochondrial membrane. And here's the matrix of the mitochondria. Okay? I got a little legend here where it says this right here is the precursor protein. And this little helical thing is an N-terminal matrix targeting signal. So N-terminal because it's on the N or the amino side of the protein. And it's a matrix targeting signal. Remember what I said a targeting signal does? It targets it to a specific place. In this case, it's called a matrix targeting signal because it's going towards the matrix. Okay? Now, how does this protein actually get to all of these little machineries and interact with them and then get threaded through and then get even more threaded towards the matrix? There's a lot of steps that seems to be going on here. Okay? And don't you worry, each of these things have a name, and we'll be talking about all of these names, okay? But I want to introduce you to a couple of things first. First things first being TOM. What is TOM? Remember I said it stands for translocase of the outer mitochondrial membrane? Well, there's also a TIM complex, okay? It stands for translocase, you guys guessed it, of the inner membrane or mitochondrial membrane, whichever you prefer, okay? Other things I want to introduce you is this concept of chaperones. Chaperones are these little proteins here, okay? These little blue proteins. We'll talk about them a bit more in a second. But chaperones are basically proteins that guide precursor proteins to where they're supposed to go, and they keep them in an unfolded state. Okay, so these are chaperones. Another thing I want to introduce you to is this symbol. Okay, what is this entire thing referring to? This specific symbol refers to a membrane potential. Membrane potential. And it's basically saying that the mitochondria, the mitochondrial membrane, has a membrane potential. And it's usually about negative 150 millivolts. Okay? Now you might be saying, hey Keon, how does it have a membrane potential? Well, you guys remember the electron transport chain, right? In the electron transport chain, we would pass electrons throughout different complexes like complex 1, complex 3, and complex 4, okay? And each of those complexes would in turn pump these protons into the intermembrane space, right? You guys would remember that. Now, what that's going to do is it's going to create an electrical and chemical gradient, okay? It's also going to create a pH, okay? So this place is going to be more acidic, okay? Now, this right here refers to a membrane potential, but really, if I were to be truly honest with you guys, it stands for something called the PMF, the proton motive force, which is actually built up of the membrane potential and the pH, okay? But that might be a bit too advanced for today. Now, either way, when I pump all of these protons into the IMS, I'm making the IMS very positive, and I'm making the matrix very negative, okay? That's going to be very important. That's going to establish an electrical gradient, okay? It's going to be important for pushing this protein inside to the matrix, okay? All right. I think that's all I wanted to talk about in this page. Just one more thing. Again, any protein that has to be imported into an organelle has to be in its primary format, okay, in its primary structure. So you know proteins have a primary, secondary, tertiary, and sometimes even quaternary structure, right? But we want them in the primary state when we actually thread them through these channels. So for example, once it gets into the matrix, it'll fold back towards this structure, okay? It'll turn into its, I don't know, probably tertiary structure here, okay? You see how it's very folded and convoluted. That would be hard to get through this channel, right? We need it to be all straight and primary, okay? So this primary structure is just a chain of amino acids. So amino acid, amino acid, amino acid, okay? Held together by peptide bonds. Okay, that's all I wanted to talk about on this slide. Now let's talk about how this protein goes from the cytosol to interact with certain proteins on the actual outer membrane, okay? So, one of the proteins that we're going to talk about first is going to be this protein called the heat shock cognate 70, okay? And HSC70 is a chaperone, okay, that has ATPase activity, meaning it's able to take ATP and cleave it into an ADP and inorganic phosphate, okay? That's good. Why does it actually do this? Well, it does this, it utilizes ATP to keep the protein in an unfolded state, okay? To keep the protein in an unfolded state. Now, there's some other proteins here like DJA1 and DJA2 and AIP. DJA1, DJA2, and AIP, these are all called co-chaperones, okay? So AIP stands for aryl hydrocarbon receptor interacting protein. You don't got to worry about the name too much, okay? But AIP actually interacts with the N-terminus matrix targeting signal, okay? And specifically on the positive charged side of it. So this NMTS, okay, it has two parts. It has a positively charged side and a hydrophobic side. You guys know hydrophobic means it hates water, okay? Which means it actually loves lipids, okay? So we call it lipophilic as well, okay? Now, this positively charged side would be polar, so it actually loves water, okay? So it loves water. We call that hydrophilic, okay? So it has a hydrophilic side and a hydrophobic side. We call a molecule that has both of those properties amphipathic, okay? A molecule that is both hydrophilic and hydrophobic is amphipathic, okay? Now, this, NMTS also has a specific structure, okay? It kind of looks like this. It's got this little, like, helices, okay? And because of that, it's called an alpha helix, okay? So sometimes you might see this be called an amphipathic alpha helix, okay? Amphipathic alpha helix. Positively charged on one side, and it's hydrophobic on the other side, okay? Now, what else do this DJA1, DJA2, and AIP do? So AIP, DJA1, and DJA2 enhance the ATPase activity of this chaperone HSC70, okay? So that's great. They actually assist HSC70 by transporting the protein through the cytosol and towards the outer membrane of the mitochondria, okay? What else does this AIP do? Like I said, this AIP, it binds to this N-terminal matrix targeting signal, specifically to the positive side of it, the positively charged side of it, okay? And this AIP is what's responsible for binding to this protein here called TOM20, okay? TOM20. And specifically, the C-terminus of this TOM20 protein is very acidic, and so that's how it binds to this AIP, okay? And I'll show you what I mean by that next slide, okay? But so that's how cytosolic chaperones bring this protein, this precursor protein, in close proximity to the TOM complex, okay? So now, how do we get from the outer mitochondrial membrane, okay, so now that we've brought this protein in close proximity to the outer mitochondrial membrane, how do we get from here to the intermembrane space? How do we get there, okay? Now, remember that I said there would be a protein here called AIP, okay? That was the co-chaperone here, okay? And of course, there was an HSC70 here and the DJA1 and DJA2, okay? I just removed them in this diagram for simplicity's sake. But this AIP is going to interact with the C-terminal side of TOM20. So this is the C-terminal side of TOM20, and this is the N-terminal side of TOM20, okay? But the C-terminal side is very, very acidic, okay? And it'll interact with this AIP, okay? And then it'll pass it on to TOM22, okay? So how does this actually happen? Well, like I said, this hydrophobic portion of the NMTS alongside this AIP bind to the C-terminus of the TOM20, okay? And then this precursor protein is going to attach towards the N-terminus of the TOM22. So here's the N-terminus of the TOM22 protein, and here's the C-terminus of the TOM22 protein. You can see that it's actually kind of protruding outwards in the intermembrane space. That's going to be important. Remember this for later. But you can see that this precursor protein is going to go ahead and interact with the N-terminal portion of this TOM22 protein, okay? And this TOM22 protein will actually sort through the precursor looking for the signal, the targeting signal, okay? Once it recognizes that signal, it'll then pass it on to this TOM40, okay? Now, you might be thinking, how does this protein just get, passed along here? And the driving force for the movement along the pathway is believed to be because of this binding chain hypothesis, okay? So first, there would be an increasing affinity of the binding site as the pre-sequence is moved throughout this pathway, okay? So maybe there's a little bit of affinity here, then there's a stronger affinity here, and there's the strongest affinity for this protein here, okay? Now, there's other factors that play a role here. So, for example, the electrical membrane potential of the mitochondria will actually play a role in pulling this protein in. We'll talk about that a bit more later. Another factor that was identified in certain papers is the fact that the cytosolic side of this TOM40 protein is very negatively charged, and the IMS side of this TOM40 protein is positively charged, which again assists on pulling this protein in, right? Because this protein has a positive charged side to it, okay? And it allows for this protein to come in electrophoretically, okay? Okay, so now just a couple of structural things about these. TOM40 is known as a dimeric beta barrel. What does that mean? So a dimer is when two proteins are attached together covalently, okay? So these are dimeric proteins, okay? One dimer here and another dimer here, okay? Great. Now, something else you have to know is that it has a beta barrel shape, okay? A beta barrel just literally refers to the fact that the protein is shaped like a barrel, okay? So that's that. It's a dimeric beta barrel structure, and it's the main passage through the outer membrane, okay? The main passage through the outer membrane. And TOM40, as you can see, right, it can actually fit two helices, two alpha helices, okay? So it can fit one here and then another one here, okay? So that's great. Now, all of these protein complexes, okay, they all require some form of stabilization to the membrane. So how is this done in case of TOM40? TOM40 is actually stabilized by TOM5, 6, 7, and a little bit by TOM22. Okay, so you can see how they're all kind of peripherally attached to TOM40, right? Here's 5, 6, 7, and 22. And again, on this side, 5, 6, 7, 5, 6, 7, and 22. Okay, these are all involved in the assembly and stabilization of TOM40. Okay, all right, so that's how we actually move this protein from the outer membrane to the intermembrane space. What happens when we're in the intermembrane space and now we want to go to the inner mitochondrial membrane, okay? We want to pass through that inner mitochondrial membrane. Well, in this case, there's a whole new complex that we come across. We come across the TIM23 complex, okay? Now, over here, there is a TIM23 and a TIM17, which actually come together and form this complex, okay? They form a subunit or a complex here called the TIM23/17, and this forms an aqueous pore that links the inner membrane space towards the matrix of the mitochondria. Okay? So that's what it does. Now, there's going to be a couple of ways that once this protein is threaded in, right? Here's our precursor protein. There's going to be a couple of ways that it can actually get towards this TIM23. Okay? There's a couple of ways, so pay attention here. It involves the interaction of TOM22, so here's TOM22, TIM23, right here, and TIM50, okay? It involves the interaction of these three giant complexes to transfer this precursor into the aqueous pore, okay? There's also going to be TIM50, which I talked about here, and at the bottom, there's going to be another protein, TIM44, but I'll touch on that later. Okay, so again, bear with me here. This process is very complex, but remember TOM22 from the last slide? So here's TOM22. Let me erase all this, okay? So here was TOM22, and I told you that this is the N terminus of it, and this is the C terminus of it, okay? And the C terminus of it kind of protrudes into the IMS. Now, if we come to this slide, you can see that the C terminus of this TOM22 will actually interact with this protein and push it along to this complex, this TIM23 and 17, okay? So these three guys will work together to move that precursor protein from the TOM complex to the TIM complex, okay? This process is actually facilitated by the electrochemical gradient here across the actual inner membrane. So how is that the case? Well, remember, the positively charged N-terminal mitochondrial targeting sequence will be electrophoretically pushed towards the negatively charged matrix, okay? So that's how this protein is going to be pushed here. TIM50 actually plays a regulatory role here. You can imagine this bulbous head that it has as a sort of cap for this TIM23. So imagine it'll literally sit on this guy and it would block it, okay? When would it do this? Well, in an absence of a precursor protein, it would actually just plop down its head and close this pore to maintain the electrochemical gradient that we established, okay? So it stops from any charges leaking in and out, all right? And it maintains the mitochondrial membrane potential, which is very crucial for not only making ATP, but protein import, okay? Now, there's other things here that actually help with the transfer, which I'm going to talk about here, okay? The protein import into the matrix, okay, is going to be a little bit more tricky, okay? The inner membrane of the mitochondria contains a special phospholipid, okay? And this special phospholipid is going to be called cardiolipin, okay? You can imagine it as one glycerol molecule with 4 fatty acids attached to it here, okay? So that's cardiolipin. Now, cardiolipin, again, it's not shown in this drawing, but it's a very special phospholipid that's only found in the inner membrane of the mitochondria, and it interacts with the protein that is currently arriving on this TIM23 and 17, and it orients it. It helps orient this precursor protein in a way that the proteins that are inside of the matrix can interact with it, okay? So it helps orient the precursor protein, okay? Now, there's going to be some other things here, such as this protein here called TIM44. TIM44 is known as an adapter protein, okay? It's an adapter protein. And all an adapter protein is that it's going to help recruit other proteins towards itself, okay? What is the protein that it's going to recruit? It's going to recruit this guy called mitochondrial HSP70, okay? HSP stands for heat shock protein, okay? So this mitochondrial heat shock protein 70 will be recruited by this adapter protein TIM44 as this precursor protein is arriving, okay? Now, so this process of binding to TIM44, or what we call latching to it, okay, is ATP dependent. Okay, so it's regulated via ATP hydrolysis, as shown right here. Okay, so when it is actually bound, it is actually hydrolyzed. It is actually um, recruiting ATP. But when it's unbound, okay, it's actually taking ATP and hydrolyzing it. So again, when it is bound to TIM44, this is the configuration that it has in regards to ADP. And when it's unbinding, this is the configuration that it has, okay? Now, in order for this mitochondrial HSP70 to actually be able to interact with this protein or this pre-sequence protein and pull it in towards the matrix, it needs to be able to be unbound from TIM44, okay? Once the process is done and the protein is fully in the matrix, mitochondrial HSP70 binds ATP again so that it can bind to TIM44, okay? So it would be in this state again, okay? Until a new protein comes along, the precursor protein comes along, and then it does this so it can separate from TIM44 and continue pulling this protein in. But how does this mitochondrial HSP70 actually pull in that protein? Well, there's two mechanisms that it uses. It can use what's known as a Brownian ratchet and a power stroke, okay? And it's depicted nicely here. So now, how do these mechanisms work? So first of all, it's proposed that these two mechanisms are not mutually exclusive and they may actually happen both together. But the Brownian ratchet basically states that multiple mitochondrial HSP70s will actually unbind from TIM44 using ATP hydrolysis, and then will search for and bind to the areas of this precursor that are unfolded, okay? And then they will pull it in, all right? Actually, rather than pulling, it lets the thing slide through their grip, okay? And this is set to prevent retrograde movement of the protein back up to the IMS, okay? So that's one of the proposed mechanisms. The second one is this power stroke mechanism, okay? And this power stroke mechanism is, again, it's proposed that upon ATP hydrolysis and the separation of mitochondrial HSP70 from TIM44, the MTHSP70 will actually change its structure and do a sort of mechanical pulling of the precursor protein into the matrix, which occurs in a step-by-step fashion, okay? So it'll pull it, and then it'll go back to its original position, and then pull it again. Now, there's many reasons why this mechanism right here is unlikely. One of the reasons is that mitochondrial HSP70 has a very small intramolecular movement, okay? Very small intramolecular movement. And so there's a very low percentage that things can be done in this stepwise fashion shown in the power stroke. And so perhaps the power stroke is kind of a secondary mechanism rather than a, actual driver. And therefore the Brownian ratchet has been said to be the main driver of this process, okay? Now, we've talked about so far how the actual protein gets pulled, but now that it's in the matrix, what actually happens? So, as soon as this protein actually reaches the matrix, okay, fully, the N-terminal matrix targeting signal will immediately be cleaved, okay? Why? Because we don't need it anymore, okay? We're not targeting that protein towards the matrix anymore, right? It's already in the matrix, so let's just cleave it and get rid of it. So that NMTS will become cleaved, and the protein will start being refolded in the matrix. Okay, how? Well, more often than not, when the protein enters, it'll be cleaved by this enzyme called mitochondrial processing peptidase, which is a metalloprotease, okay? So it's a metalloprotease. And for you guys who don't know, a metalloprotease basically requires a metal ion for a cofactor, okay, in order for it to work. So, a common one being a zinc metalloprotease in the mitochondria, okay? Now, sometimes there's another enzyme that needs to be acted on here, okay? And that's the MIP, and that's called the mitochondrial intermediate peptidase, okay? So sometimes one cleavage is not enough, and we actually need to make a second cleavage, okay? And as you can see, in certain examples, like this POTC here, this stands for precursor of ornithine trans carboamylase, okay? And this is an enzyme that's actually used in the urea cycle, okay? If you guys remember, your urea cycle actually takes carbamylate and combines it with ornithine to give you citrulline, okay? And the enzyme there is ornithine transcarbamylase, okay? So again, most proteins, as soon as they come in, their NMTS will become cleaved by the mitochondrial processing peptidase. However, certain proteins, such as this precursor of the ornithine transcarbamylase, requires two-step breakdown, in which the first cleavage is done by the matrix, the mitochondrial processing peptidase, and the second one is done by this MIP. Okay. After which, what happens to this? So here it is. Here's that protein that we had, right? Here is the actual protein, and here's the NMTS. That NMTS will, go off and get degraded, okay, by the mitochondrial's intrinsic degrading system. And this protein will become folded. Sometimes this protein can become refolded in and of itself. Other times, and majority of the times, it actually needs to go through a folding machinery, which consists of HSP60 and HSP10. Sometimes you might also see them called CPN60/CPN10. Okay, but this process requires ATP. Okay, and what's really actually happening is that HSP60 is the one that has the ATPase activity, and it is thought that this protein here, HSP10, regulates HSP60's activity. Now, this is a more schematic representation of it. This seems to be a bit of a more accurate representation of it. The HSP60 is a two-ringed heptameric, okay, heptameric region, okay, and this two-ringed heptameric region at either end is going to be capped by an HSP10 heptamer, which creates an isolated environment and then prevents aggregation of the protein with other things in the matrix and promotes a state of folding. Okay, so this protein is going to go inside of this cavity of the HSP60, and on either end, it'll get capped by this HSP10, which creates a isolated environment for folding, okay? And again, it's a two-ringed heptamer, hepta like 7, okay? So there's 7 sort of, you can call it subunits, okay? Capped at either end by this HSP10, okay? And once that happens, this protein will finally become folded and then, move on to do whatever the function of it was, okay? So just to clearly cap everything together, here's what the whole system is going to look like altogether. So first of all, we're going to have this precursor protein, okay? And this precursor protein is going to have an N-terminal matrix targeting signal, okay? Which is basically a sequence of amino acids that says, hey, I need you to go towards the matrix, okay? Great. Now, it has a positively charged side, okay, which is hydrophilic, and it has a hydrophobic side, which is lipophilic, okay? This protein is kept in this unfolded primary structure by the help of HSC70, a heat shock cognate 70, which is a chaperone, okay? It's basically a chauffeur that guides the protein to where it needs to go, okay? And there's other things like co-chaperones, like DJA1 and DJA2 and AIP, which regulate the ATPase function of HSC70, right? So remember, this process requires ATP hydrolysis to keep the protein in an unfolded state. Now, remember I told you that through the binding chain hypothesis, this protein will get pushed along the Tom complex, okay? So first, the AIP will actually bind to the C terminus of this Tom20. So right here is the C terminus of the Tom20. right? And the hydrophobic region of this NMTS alongside the AIP bound region will bind to this TOM20, and then it'll get pushed towards the TOM22, which sorts through this protein looking for that targeting signal. Once it finds that targeting signal, it pushes it down to this dimeric beta barrel protein, TOM40. TOM40 is that main passage from the cytosol to the IMS. Once this protein gets threaded through towards the IMS, it goes through a series of interactions where the cytosolic C-terminus of TOM22 interacts with TOM50 and the TIM2317 complex to transfer this pre-sequence from the TOM complex all the way to the TIM complex. Okay? Now, this process is further facilitated by this membrane potential. Remember this membrane potential? I said it was created by the electron transport chain, which we pumped a whole bunch of protons here, making the IMS more positive and the matrix more negative. Well, that's going to attract this positively charged protein all the way into the matrix, which is negative. Okay? I also said that this process here is regulated by TIM50. TIM50, you can think of this giant head that it has as a sort of cap, where in the absence of a pre-sequenced protein, it kind of just puts his head down and regulates the membrane potential, not allowing any leaks to go out or in. Okay? Now, how does this protein get pulled through the TIM2317? Well, I told you that there's a TIM44 adapter protein here, bound right towards the membrane. And I also said that there's a special phospholipid that resides in the inner membrane called the cardiolipin. And cardiolipin helps orient this precursor protein in a way where it can interact with these guys. Okay? Once this protein comes through, TIM44 recruits mitochondrial HSP70. Okay? And when it's bound, it has an ATP attached to it. But when it unbinds, it hydrolyzes that ATP into ADP and an inorganic phosphate. Okay? That is when it's going to go through that Brownian mechanism. Okay? Remember we said there was two mechanisms. We said there was going to be a power stroke and a Brownian ratchet, which we said the Brownian ratchet involves multiple mitochondrial HSP70s coming over here and kind of gripping onto areas of the protein that are unfolded, basically allowing for it to slide through its grip into the matrix and then stopping retrograde movement back into the IMS. Okay? And again, we said that process requires ATP. Next, we said that once this protein comes in, the N-terminal matrix targeting signal will immediately get cleaved off by the mitochondrial processing peptidase, and sometimes if the protein is large enough, such as the precursor version of ornithine transcarbamylase, you're going to require a second cleavage, which is done by the mitochondrial intermediate peptidase. Okay? And so here you can see that the peptidase will do its job and get rid of this alpha helix, and you're left with the protein. The protein will then go and interact with HSP60 and HSP10. HSP60 has a ATPase activity, okay, and it is thought that HSP10 regulates the ATPase activity of HSP60. Now, what's going to happen is this protein will go inside of the HSP60, and this HSP60 actually has, like I said, a heptameric region, okay? It's two heptamers that are bound together, okay? So there's one heptamer, here's another heptamer, bound together, and they create an isolated environment for the protein to reside in. And this environment is capped at either end by our HSP10, okay? Again, further creating an isolated environment and promoting a folding of this protein. And then, of course, this protein will go out, and now it's in its folded state. Here's the carboxy side, here's the amino side, and it's properly folded, and now it can do its function. Maybe this protein is related to the electron transport chain, where then it will join there. Or maybe it's some sort of transcription factor that's involved in mitochondrial biogenesis, for example, TFAM. Okay? But that's going to conclude protein import into the mammalian mitochondrial matrix. Okay? Again, if you guys benefit from this, please follow and, like, leave a comment. I'll see you next time.